Well, thanks everyone for coming. This is it's really refreshing to see as much attendance as we've gotten, and to really work with the proactive lake association that we have. And I really have to tip my hat to Dan for kind of sparking this new energy and the interest into AIS, and really, he really has been the spark plug behind getting this grant that we have for AIS and this new portion of our monitoring. We first sampled here in 1999 and 2000. It wasn't me that came up those years, it was the principal of our company. And as I believe it was in response to a proposed subdivision that was up here. A lot of work hadn't been done in the area. They said, well, what is the status of the lake? What kind of water quality do we have? And what kind of impacts could happen if this subdivision goes through? So what we did was kind of show like what kind of impacts can come from a new subdivision, from septics. We did some modeling to show that these are the potential nutrient inputs that can happen from the density of homes that they were proposing, and that kind of helped steer management decisions toward whether or not they were going to approve this subdivision. And ever since then, the Lake Association is really involved with water quality and the sampling that we do every year. We've done yearly sampling since 2004, and I've personally been coming here. I think I first came in 2008 or 2009, so it's been about 10 years that I've been coming up. We typically sample in August or September. The last few years it's been the first week of August. I try to time it in conjunction with the annual Lake Association meeting. That way I'm, last few years I've kind of given a presentation at the Lake Association meeting in August, and that way I'm available to answer questions and whatnot. But it's also a good time to be consistent. August is a time of year where the lake is typically warmest. We're probably most likely to see algae blooms, and just getting year by year consistency with that kind of timing, we're able to look at trends over different years. And we also sometimes come here in spring. I've sampled, I think, 2 times in the spring before, and part of our grant this year, I proposed to sample this year in the spring, and one reason was just that it's a high water year. We had about 150% snowpack this winter, and one thing that you can get with high snowpack and increased stream flow is just an additional flushing of nutrients out of the watershed. And one thing that our data has shown is that on years with high water, we do get a little bit of a spike in sediment and in nutrient input to the lake. And we see that especially in 2011. It was a year where the nutrient input was a little bit elevated, and that's just more nutrients that are available for plant and algae growth. And our sampling that we do, it primarily is focused around nutrients. Tyler talked a lot about them. I'll talk a little more, but nitrogen and phosphorus are the 2 main nutrients that control plant growth. And plant and algae growth are what we refer to as primary productivity. And they photosynthesize, they consume nutrients, and they kind of create the basis of the food web. And fish primarily eat aquatic insects. They eat, typically the larvae they typically eat bugs is what we call them. They're little mayflies or stoneflies, or their larvae. And they don't necessarily eat algae. But one thing that algae does, it can consume oxygen from the environment. So that's one reason we look at nutrients, is that nutrients feed algae, and algae kind of competes with oxygen with aquatic invertebrates and with fish. So a lot of people say, well, why do you sample for this? And that's really the primary reason why. It creates an oxygen demand that can make it hard for aquatic invertebrates to survive, and it can make it hard for fish to survive. So as I mentioned, the algae in the plants, they're the primary producers. They photosynthesize during the day and they produce oxygen. And at night they actually respire and they consume oxygen. So the most likely time that you're going to see low oxygen in a lake or a river is right before the sun comes up in the morning. So that's not typically when we're out sampling. So sometimes if you want to find out the worst case scenario for a lake, you got to be out there before the sun comes up to see what your oxygen levels are. But we fortunately don't experience low oxygen here. All of our oxygen is high enough that we can support the fish populations and the aquatic invertebrates. But we want to make sure that we don't get enough algae and plant growth that it is a detriment to the species that we do want here. And he showed a little bit of the data that we do collect. There were a few of these handouts out in back, and if you didn't get one, I can certainly provide it for you. It's also available in our annual report. I did hand out a few copies of our annual report. If not, if you didn't get one, it's available on our website. So go to the website and there'll always be the latest report. I don't think the 2017 report is on there yet, but once it's done, we'll get the 2017 annual report up. But this does show that we have seen a declining trend in nutrients. Nitrogen and phosphorus has kind of steadily gone down since 2004. We do get kind of spikes here and there. 2011 was the one year I mentioned that we did see elevated concentrations. And Tyler also talked about this Carlson's Trophic Index score, and that's a way that we can kind of evaluate whether the levels of nutrients that we're seeing are potentially hazardous or could potentially turn into problems at the lake. And all the blue dots along the bottom here, that's nitrogen. And nitrogen is typically higher than phosphorus. And what Tyler mentioned is that this lake is what we call phosphorus limited. Nutrients come from a lot of different sources. There's natural background sources, but it's also tied with sediment. It comes from decay of vegetation, especially septic systems are probably one of our biggest sources of nitrogen around here, especially failing septic systems or older systems that are contributing to the lake. And fertilizer is another source. So, you know, like he said, if you're fertilizing right up to the bank of the stream and any of that is getting in there, you know, that's a potential source of nutrients. But we are, as you mentioned, phosphorus limited, and what that means is that plants, they consume nitrogen and phosphorus in different ratios, and I believe the ratio we typically refer to is 15 to 1. They typically take in 15 times as much nitrogen as they do phosphorus. But we have enough nitrogen in the system to feed plant growth and algae growth. We're more likely to see an increase in algae if we increase the phosphorus because it's limited in the environment. So that's why we use that term. And phosphorus is something that's more associated with sediment input. So anytime there's new building or any kind of road building, anything that's going to be clearing vegetation and increasing the amount of sediment into the lake, that's a potential for increasing phosphorus. And also just sources from the upper watershed. There was a timber sale in Upper Herrick Creek within the last couple of years. They did not only logging but also slash burning, and that can be a source of nutrients. So you know, anything in the upper watershed could act as a source of sediment. We do see limited algae growth in the streams. There was this handout back there too. If you didn't get a copy, we can get one to you. But we typically see the highest concentrations of algae at depth, especially in August. And we a lot of times see it even like 60 feet down. And part of the reason is the nutrients in the upper water column are being depleted by the algae. So the most favorable spot for them to grow is further down in the water column. So this data here, this is the lake profile data. Tyler shared some of this, but it shows kind of the stratification that we have of the lake. The upper 30 feet, this is temperature up here. The upper 30 feet is typically really well mixed. And then we get this area of temperature decrease called the thermocline. And then once you get to about 80 feet or so, it's all really cold water and it's all not very well mixed. But these profiles change throughout the year. We come back every August and do this just for consistency, but times of the year the lake will be completely well mixed and you'll have the same temperature top to bottom. We also get stratification with oxygen concentrations too, and some of that has to do with the algae in the lake. The algae is a primary producer. It produces oxygen during the day, so we'll see higher oxygen at depth. And you do see changes with pH too, and that also kind of ties into some of the algae and plant growth that goes on. And we have done additional sampling beyond just the nutrient stuff that we do. With nutrients, we do the inlet stream and the outlet stream. We do the center of the lake, and then we do 6 additional samples that kind of bounce around the lake. And that's just to see if we can identify any kind of spatial differences around the lake. You might get groundwater input in different sides that change water chemistry. So you don't see a whole lot of variability around the lake, but if there is, we want to capture it with our water sampling. And year to year, different topics may come up. I mentioned that there had been like a timber sale in the watershed a few years ago, so we kind of added a few different variables or a few sites in the upper Herod Creek, just to get baseline data, just to show that this is what the conditions are now. If they change in the future, at least we have the data to kind of fall back on. And also, some of you might be familiar with in the community of Kyla, they had kind of a scare with radioactive materials. There were radon detected in a lot of wells around Kyla, and people around here were like, well gosh, Kyla's right down the road, and is this something we have to worry about? Do we have to worry about radon in our wells? So we added uranium and radon a couple years ago, and we sampled not only in the lake but in groundwater just to see if it was a concern. And fortunately, everything came out really low. Most of it was all below detection. So as new topics emerge year to year, we're able to add those to our water quality program. And the water sampling has been funded through a DEQ volunteer monitoring grant. So we've put in for that the last 4 or 5 years or so. So all of the laboratory analysis is funded through this DEQ volunteer monitoring grant, and they've been really great. They like how proactive we are up here, and the fact that we have this lake association that wants to do something about it, and they're really supportive. So every year they've always funded everything we've requested, So, and that's kind of a thing for you guys to think of too. If there are topics that kind of come up where you're like, hey, what about this? Have we ever sampled for this? Is this something we need to be concerned about? We can certainly kind of consider it and, possibly add new sample sites or new parameters to sample for in the future. So always keep in mind, other topics that come up, especially related to water quality. And Dan talked a lot about the DNRC AIS grant that we put in for this year, and it was kind of a whirlwind week when we first put in for it. The Lake Association decided, yeah, we want to go for this. We want to, see what we can do for AIS this year. But like you said, we couldn't. The grant had to go through some kind of government entity, and we thought, well, we kind of kicked around a few different ideas of who we could rope into it, and the best option was to just focus as close to the lake as possible, and that was the school. And we thought, well, if we're going to get the school involved, we've got to get an education component, we've got to get the kids involved, and we want to include them as much as possible with this program. So I came up here, I believe it was in March, and we forced our way into a meeting with Sherry, and said, yeah, it was like, we're coming, we're going to talk about this idea, hopefully you're on board with us. And luckily they were, and the school board supported our ideas too. And I went back to Butte and frantically wrote a grant application, and luckily we were successful at getting the $10,000 this year. And as Dan said, there were 15 applicants, and they approved 8 of them, and we ranked 5th out of the 15. So I think it was kind of cool that we were able to get the grant this year. It's an off year for the grant cycle, so they didn't have as much funding as they normally do. But the fact that we kind of pushed it through the door and we were able to get funding this year, I think was awesome. And there are 2 main components of it. We're going to have a monitoring and sampling component to it, as well as the education and outreach. And the monitoring component of it is primarily going to be based around vegetation and mussel sampling. They've done pretty limited vegetation and mussel sampling in previous years, so we said, hey, we want to do a more exhaustive survey of vegetation. We're going to do a full shoreline survey, identify what plants are out there, and also try to identify if there are invasive species anywhere around the lake. And we're going to do mussel sampling as well. Fish, Wildlife, and Parks does some mussel sampling, and I said, is there any harm in duplicating your guys' efforts? And they said no. Looking for mussel villagers is kind of like hunting for a needle in a haystack. So they said the more samples you guys can collect, the better chance we have at detecting them. So we've ordered a plankton tow net. It basically looks like a pantyhose hooked to a wire, And there's two types of samples you do. You can do a horizontal tow, where you drag it behind a boat for a certain distance, and collect that in a bottle. We send it off to a lab where they analyze it. And we're going to do a microscopic analysis, and we're also going to try to get an eDNA sample where they look for the muscle DNA. And we also do a vertical tow sample where they drop it down and collect a vertical, through the water column. So we're going to do two types of muscle sampling as well as that vegetation survey around the lake. So, and on top of that, we're also going to do the usual water quality sampling that we're doing, all the nutrients that we've done, but we've also added parameters that are kind of more specific to aquatic invasive species, such as calcium and alkalinity and hardness. And those are really good because it's an indicator of potential colonization. The mussels build their shells out of calcium and magnesium, so they need a certain level within the water of hardness in the water to build their shells. So we'll sample a bunch of different sites around the lake and see what the potential for colonization is. And the education program, I think, is kind of the coolest part of this. We're going to get the kids involved with the program that we're already doing. We're already out there doing this sampling anyway. We want to reach, extend our data and our information out to as many people as possible. And also, the way we look at it is the school kids are kind of like the next generation of lake users. And the sooner they learn this information, the better off they're going to be in the future. So I think it's great that we got the school involved. We're going to, as mentioned, try to do field trips this fall. Part of the funding will go towards transportation just to get them on a bus, get them to the Flathead Biostation. We also want to get involved with the science fairs. Flathead County has a great science fair. I believe it's going to be in February of next year. So we're hoping to get a few kids involved. We can work with them in conjunction with what we're already doing. We're already doing a vegetation survey. We're already doing mussel sampling. We're already doing water quality sampling. So any one of those things, if there's a student that wants to get involved, send them our way, and we can act as a mentor. We can turn our existing sampling into some kind of a science fair project and mentor them, help them interpret the data and put together a poster and a presentation. And one thing Dan has talked about too is how he wants to kind of make the Marion school kind of like the lake school. We want to have this a school that acts as a template for other places where they can see what we're doing at this school and hopefully look at it and say, hey, this is great. We have this great classroom right in our back door being the lake. So why not get them out there and get them involved? But also the people who are applying for these DNRC grants are typically, we're definitely the only school that's applied for it. So later this year I'm going to present some of the work we've done at conferences just to show Marion School as kind of a template of what schools can do to get funding, to do monitoring, and to also just get the kids involved and kind of outreach to the community. So, but also this is our first year of this DNRC AIS grant, and we hope we're planting the seed for the future. This year we weren't able to fund inspection stations or a decontamination station, but the fact that we have volunteers who are willing to go out there and educate people and kind of act as pseudo inspectors, I think if we can show the DNRC that we have this much involvement already, I think they're going to be more likely to fund a station in the future. So that's where we're looking for future years, not only the monitoring and education, but also getting inspections and decontamination. So I did bring some tools of the trade up here. I want to kind of just show you some of the stuff that we do. So I'll probably have to drop the mic here, but I'm usually loud enough people can hear me. So with water quality sampling, there's usually not a lot to it. Typically all we're doing is just going around and filling bottles. We collect samples for a lot of different things, nitrogen, phosphorus, sediment. We also sample for chlorophyll A. Chlorophyll A is a pigment that's produced by plants and algae. So chlorophyll A acts as an indicator of algae growth. So we go around and fill about a half dozen or so bottles at each of these sites. Some of them have to get filtered, some of them don't.
We also sample for what's called benthic algae, and that's algae that's attached to rocks. As algae attaches itself to the rocks, it can really take away habitat from aquatic invertebrates that are desirable, the mayflies and the stoneflies that are out there. So some people noted several years ago, like, hey, we're seeing more of this green slime on our rocks. You know, is this going to be an issue? And it's like, well, we've never sampled for it before. Let's go out and at least collect a baseline of data. Let's see what the levels are out there. Let's see if they are approaching what they consider nuisance levels. And if we do this year to year, we'll see if they're increasing. But benthic algae sampling is kind of interesting. It's usually just you find rocks with algae on it, you put a template on there, and you scrape off the algae within that known area. And then we take that and we filter it through this unit, and then we take that filter and send that to the lab, and they were able to extract the chlorophyll off of that filter and tell you this is the concentration of chlorophyll per unit area. So usually they report, like, for every meter squared you have this much chlorophyll, and you can compare that against other data. You know, a lake that's, you know, really infected might have 100 micrograms per liter or per meter squared of chlorophyll A, but our lake is really low. A lot of times we can't even detect it. They're at such low levels. And this is a water quality meter that we use at each site. This gives us field data that we collect at each site. Mostly temperature, pH, specific conductivity, and oxygen is a big one we like to look at in the lake. But you know, he always says, you know, when you go to the doctor and you get your blood pressure and they take your height and weight, I always think that this is kind of like the blood pressure and the height and weight and that of a lake. You got to come out and get the basic parameters. You got to find out what's the temperature, what's the pH, what's the conductivity, what's the oxygen in the lake. So we do that at each site. This is a depth sampler that we use. I believe this is called a Kemmerer sampler. I just triggered it there. This thing's got a really sensitive trigger. But this is what we use to sample the lake at depth. And hopefully I'll be able to get a demo here with you. But this guy here, we a lot of times we'll want to target specific areas of the lake to sample rather than just do grab samples right off of the surface. As we showed in those profiles, the water chemistry changes with depth, so we don't always just want to get stuff from the top. We use this for targeting stuff at lower depths. So we usually take about 3 or 4 different samples at depth. And the way this works is you lower it down where you like it, and it's got this this guy here called the messenger. You send down the messenger, and it triggers it, and then that captures the water once you're down there. And you can collect a sample off of this. So my kids always like playing with this and snapping their fingers. And this is a Secchi disc. This is probably the oldest thing they've used in lake research. It's basically used for looking at water clarity. And we have a really clear lake out here. I think what you do is basically just lower this down until you can't see it anymore. And then you bring it back up and you record where you can see it again. And we usually do that about 3 times or so. You get where does it disappear, what depth does it reappear, and you can get an average. And we typically have a Secchi disc depth of about 40 to 50 feet, which is really good. So we have a really clear lake. It's a good indicator of good water clarity. So we do this every summer. And there's a program called the Great Secchi Dip-In. And does that take place in August or? Yeah, I think it's August, but it's one day out of the year where they try to get all these lake associations and stuff to go out and get a Secchi disc depth on their lake, and then they send that data in somewhere, and you can compare it to lakes all around the region and all around the U.S. So the Great Secchi Dip-In happens in August, and it's kind of one day a year where everybody goes out and samples their lake. And this is a vegetation rake. This is what we're going to use later in the year to help sample vegetation. Most of the aquatic vegetation doesn't grow below 25 feet. So I handed out these are in the back room there, but this is a bathymetric map of the lake, and Bathymetry is just a fancy word for meaning the lake depth. So it shows like the depth contours of the lake. Little Bitter Lake is 260 feet deep at its deepest, right out in the center. And what this shows, the shaded zone here that looks like vegetation, that's what's called the littoral zone, and that's less than 20 feet. And I wanted to hand this map out to you guys because these are the areas where you're most likely to see aquatic vegetation, or where you're most likely to see aquatic invasive species. So typically we don't have a lot of littoral habitat on the east side of the lake. It's pretty steep sided, so you know, we don't have a lot of shoreline habitat, simply put. But most of the shoreline habitat is kind of here on the southwest part of the lake, and definitely in that northwest bay. Sometimes that northwest bay will completely fill up with vegetation. So when we come out and do our survey, we will do we'll cover the whole circumference of the lake, but we're also going to definitely target these areas where we're more likely to see vegetation, and definitely up in this northwest bay here. And I included here on the back kind of how to identify the common invasive species. And these are the big three that we worry the most about: Eurasian water milfoil, curly leaf pondweed, and flowering rush. And these three are, I talked a little bit about Noxon Reservoir. These are very prolific nearby in Noxon Reservoir. Eurasian water milfoil, if you see it, the best way to identify it is it has what's called whorls. So the leaves kind of grow in a circular pattern around the stems, and they almost look like feathers. And if you count the leaflets that are on these feathers, there's typically about 12 or 14 in Eurasian water milfoil. So it looks really feathery. There's a native milfoil that only has 7 sets of these leaflets. So it looks a lot more coarse. So if you can't really tell, like, is this Eurasian water milfoil or is this a native milfoil, you can physically pull one out and just count the number of leaflets. And if you have over 10, you're probably Eurasian water milfoil. If you're less than 10, you're probably the native stuff. They do hybridize, so there's a hybrid milfoil that, you know, usually has somewhere in between 7 or 14. And it's hard to, I think you probably have to do genetic analysis to determine if it's a hybrid or not. But if you see something like this, I also kind of made a note down there on the bottom, what to do if you spot an aquatic invasive species. Collect a sample, you know, put it in a bag, bring it to somebody, or look online, see if you can identify what it is. Know your location where you found it. You know, we want to go back and see if we can see if there's more of it, or if it was just some kind of a fluke. You can contact your local fisheries office. There's a phone number there, or there's also on the FWP AIS website, they had a link where you can actually report AIS, and it brings you to this FW Fish email where you can report aquatic invasive species. And as mentioned, if you see contaminated boats or trailer, that 1-800-TIP-MOND is the place to call. But the other one we see around here is that curly leaf pondweed. The best way to identify that is lasagna noodles. The leaves look just like lasagna noodles. In flowering rush, we have a lot of native rush around here, but flowering rush is an invasive one. And the best way is typically to look at the flowers. It has these umbrella-shaped clusters with 3 whitish pink petals. So these are kind of the main 3 that we're going to be looking for later this summer, but we'll also look at what native species are out there. And yeah, what we do is a lot of times just drive around the lake and look visually, and every so often you have to throw this down there, and this just grabs the vegetation, you pull it up, you know, pull all the different species off it, and try to identify them. But anyway, that's kind of a bit of a background of where we've been with the lake. The lake's always had really good water quality. The Northwest Volunteer Monitoring Program, they sample like 41 lakes around the region. And we're, in terms of water quality, we're typically kind of in the top 5 or so. So I think we've really gone a long way toward protecting this lake and maintaining the water quality, and Scrape being a part of this lake association that, you know, has a passion for protecting it and is so proactive. So
Anyway, that's about all I had to talk about. If anybody has questions about what we do or what results we've seen in the past.
So if you were a board member on Little Bitter Lake Association, and you wanted to know, what the heck are you doing? What am I supposed to look for? What would be the one paragraph or two paragraphs you'd give us so that we would know when we come to Little Bitter Lake meetings, how goes it? Would you say, focus on nitrogen or phosphorus, or what would you, how would you translate all this into the simplest thing to watch for?
Well, in terms of, like residents around the lake, the biggest thing is gonna be algae growth. You guys are gonna notice if you have an algae bloom. You're gonna notice if you have more of the green slime hanging on the rocks. And it's just those kind of visual observations that have kind of, prompted us to do some of the sampling that we've had. In fact, we had a lake resident a couple years ago. He said, hey, there's this giant patch of green moss that grows out in our lake. Can you come take a look at it? And I went to his house and we got on his jet ski and he took me out there. And you don't see a lot of algae in this lake. You really don't. You don't see algae blooms very often, and even like the green moss and slime that you see around your boat docks, there's not a whole lot of it. But he took me out to this patch, and I was telling you about it yesterday. It's about 30 feet wide by 60 feet long, and it was like algae growing like that thick. And he's just like, what's going on out there? And I kind of scratched my head at it, like, I don't know what's going on out there, but one potential thing that we talked about, there was a home pretty kind of nearby, and there was a little swale kind of near the home, and we speculated that it's possible that there was leakage from their drain field that's getting into some, possibly some channel or something, some underground channel that's just delivering nutrient-rich groundwater to that patch of the lake. And it's a really isolated spot of it, but boy, the algae growth was just off the charts. So just kind of visual stuff like that you guys can see. If you're seeing an increase in algae, that's going to kind of be an indicator of something. Yeah, and that was kind of a freak occurrence. We never could figure out why exactly the algae was growing in that one spot. And we want to make sure that it's not like spreading beyond that spot, but it was kind of a... So we get a lot of feedback from residents around here, and like with the radon issue, they're hearing what's happening in the area, like, hey, Kyla has high radon. Do we have high radon? Like, well, nobody's ever sampled for it. Let's sample for it. So just staying abreast with kind of local issues and visual observations can kind of help direct us how to do the sampling. The basic nitrogen and phosphorus, we're probably always going to do that because those are the two kind of primary nutrients that feed everything. So we'll always do the basic kind of water chemistry stuff, but additional stuff we're always happy to add that on when we can. Yeah?
So the lake was down for quite a few years, and the lodgepole grew up around the lake. It was pretty fierce. And of course the tribe kept the water level down for their irrigation.
Yeah.
And then the water table came up and it drowned all of those out, and they all died. And in some areas the ice pushed them up. So what effect does all of those dead trees have on the nitrogen? You talk about logging, but this is right along the lake, and that's gonna dump a lot of nitrogen in there.
Yeah.
Correct?
Yeah, absolutely. And
And that was about 2011.
2011 to the lake, because it was such a high water.
That's when they started to die off, and they were starting to get uprooted.
Yeah, so, and lake level has been an issue that other board members have brought up a lot. And you guys, as I understand, have no control over it. It's controlled by the tribe because of irrigation. Yeah. So yeah, in 2011, like you said, the water level came up and drowned out that. That is going to be an outside source of nutrients. It's also going to be an outside source of carbon. One thing that happens is you get not only like biological oxygen demand, but you get what we call chemical oxygen demand. So as you get additional vegetation coming into the lake and decaying, that's an oxygen, a source of oxygen depletion, I guess I should say. So as that decays, it's not only going to contribute nutrients,
it's gonna contribute carbon, and it can help, and it can contribute to the depletion of oxygen, so,
Because I always hear it's always logging,
yeah.
and where I live, used to be a sawmill, they used to float logs over there and cut logs up there.
Yeah,
And it's like, and the lake was great, it's always been great.
yeah,
But I think there's a lot of other things. Don't get me wrong, I think what you guys are doing is wonderful. I like this. But I think all of those trees that died around the lake contributed to a lot.
no, I believe that, and that's something for me to consider too, as we interpret the data. Like, are we gonna, post 2011, are we gonna start seeing an upswing in either organic carbon or nitrogen or phosphorus? So, it's, and hearing that kind of feedback from you guys helps me interpret the data too, because if we do see some kind of trends like that. And yeah, and I don't mean to pick on logging either, it's a big part of the economy here, and we all depend on it too. But that's, the road building, a lot of times, it's not necessarily the clear cutting, it's sometimes the road building is an input of sediment. And for the most part, they're doing a good job. I visited the upper watershed last year, and a lot of it's pretty well disconnected from the stream and stuff like that. So it's, I think sometimes it's good to just, let's get the baseline data so that if there are changes, we have the numbers to see that, yeah, before something happens.
And then look outside of that.
And then look outside of it, yeah.
Correct. I agree.
But that's one thing that's cool about lakes, is lakes are like the bottom end of a watershed, and the water quality, within that lake is a reflection of what's going on around it and the upper watershed around it. So that's, my background's in watershed science, and that's why I like working with lakes, is that here's this point that we can look at the water chemistry and then kind of like work, outward from there. Not only right around the shoreline, but then the homes around it, the roads, and then into the greater forest and beyond, so. But yeah, I think just staying abreast with, changes within the watershed, whether it is water levels or, any kind of activities like that. And that's one thing we, I mentioned I've only sampled the spring twice before. And this year, knowing that it was such a high water year, I added June sampling. So actually, as soon as we're done today, I'm gonna go around and do some sampling, just so that we kind of have this spring data. This is when we get that flush of nutrients and sediment typically into here. So we might not see high nutrient concentrations later in the year, but we might see it more so in the spring, and that's going to contribute to algae and plant growth later in the year. So
Don't you expect that though, with the high runoff that we've had this year?
yeah, I do.
I mean, that's a normal thing, right?
Yep.
Okay.
Yeah. Any other questions? Or?
Thanks, John.
Yeah. Thank you.
So, what I learned from that is that, just like, if you have something going on wrong with you, often your skin shows it first. Skin's real sensitive to, so that algae growth, I thought of as, a lake skin irritation caused by something. So, like I mentioned, this is more than just invasive species. This is, the volunteer program is also. And now, if you're not at the boat dock talking to people, you're just riding around in your boat, clockwise or counterclockwise, big decision. You can inspect your lake. You give your lake an inspection. You can look for these things that maybe otherwise wasn't on our radar screen. You can look for patches of algae, if you will, or weeds, if you will. We've got a map now that we can look. We'll do a better one here shortly when he does the Aquatic Invasive Species.